Hey everyone, and welcome to the microanatomy section of your skeletal muscle lectures. Before I get going, I just want to try to make it clear that this lecture will focus specifically on the structures found in the sarcomere, the muscle at a microscopic level. That being said, this is a really crucial stepping stone to comprehend, understand, and at the very least, picture in your mind clearly before we get into the how do muscles work component, which will be coming up. So the explaining of how muscles actively contract, and we'll go through the sliding filament theory that will explain that. But again, this lecture really provides a foundation for that discussion that we'll have coming up. Okay. So let's dive into it. Just some introduction into some terms before we see an image here. But the sarcomere is the basic unit or section of muscle. And it's truly the component that produces force when muscles contract or shorten. What we'll see, and I've mentioned this before in a previous lecture, is I called skeletal muscle striated and multinucleated. So multinucleated just means lots of nuclei can be found on skeletal muscle. But the striated component speaks more to the microscopic structure that we can actually see from a bit more of a macro level to a certain degree. And what it is a part of the components of the protein structures are thicker and others are a bit more thin, so it gives us lighter or darker appearance. And you'll notice there's some lines and zones and bands, etc. I think these are best to see visually to understand. We'll start going through this more elaborate diagram up here, and there's two components of particular interest when we're talking about a sarcomere that you should become really familiar with. One is myosin, so this blue thick filament of proteins here in the middle. So that's myosin, and we can find that here in this image of a sarcomere. And actin, okay, which is this red component here, staggered on either side of the myosin. And so actin is also called the thin filament. So we got a thick myosin filament and a thin actin filament here, okay? And again, these are strands of proteins, okay? So again, we're not going to dive into the how necessarily, but what will happen is the myosin will pull with these little globular heads here along the actin, and that's how it pulls the cell or causes a contraction or generates force, okay? Just to give you a little bit of a heads up about that. But one of the ways we can describe that contraction or pulling together is by landmarking and indicating some other structures in the sarcomere and then describing how they move relative to one another. Okay, so if we look at the green lines, we call these Z discs, and you can see there's kind of this alternating protein structural strand that goes back and forth, and we can essentially call that the border of the sarcomere. So Z disc to Z disc will be the length or landmark borders of a sarcomere. And then we have our thin and our thick filaments here, our actin and myosin, and our M line or M band area that runs just down the middle. Again, you can landmark these on the image of an actual sarcomere as well. Okay, and so from those landmarks with these protein strands in between, and we can see myosin is attached to the M line but not attached to the Z disc. And again, if we flip that around, actin is attached to the Z disc and myosin is not. Okay. And so again, if we return back to these zones, so a zone is essentially space between a landmark and one of these proteins. So we see the H zone is this area or distance from the end of one actin strand on that's attached to one Z disc relative to the other Z disc, okay? If we look at these bands, we'll notice that the band of myosin goes from one tip through the M line to the other side. So again, this is within one sarcomere. And the I band actually stretches outside of the sarcomere. So it includes the point where the myosin ends and runs through the Z disc to the other sarcomere over here to the other point of the myosin over in this sarcomere. Okay, so I bands go between sarcomeres, and A bands are within. Okay, so they track where the myosin is. Okay, so this might seem a little irrelevant or abstract at the moment, but let's now put it into context of an actual muscle contraction. Okay, so what we'll see here is our actin, so the red lines, and again, you kind of need to picture the myosin in between the actin here. And what will happen is this H zone, as the myosin pulls, and I usually say it's kind of like a canoe. These are like canoe paddles on the myosin here, will pull the two actin filaments together. Okay, so what you'll see is the H zone will start to disappear. Okay, why? Well, these actin filaments get pulled and pulled and pulled and pulled to the point where they have nowhere else to go. Okay, so again, this actin ends up touching that actin at the M line. Okay, so that's the H zone. Another way we can describe that same motion is the Z discs get pulled closer to the M band, right? So as, again, the myosin, these blue stripes, pull the red lines in to contract, they get closer together, and the Z discs, again, get closer together, okay? Another anchor point that you might want to put in your mind is this M line. Okay, we center that, that's our anchor point, and the myosin doesn't really move anywhere. It's the Z disc and the actin that move relative to the M line. Okay, so we could say the A band doesn't ever change length, okay? But as the muscle contracts, again, this is the I band, which is between two sarcomeres, will get shorter. And the reason for that is this myosin ends up getting closer and closer to the Z disc. Right, so again, actin, myosin. As it contracts, this is the Z line here. As it contracts, the myosin gets closer and closer and closer to the point where, let's say, it reaches the Z disc. The I band now, since it attaches from that end of the myosin to the other end of the myosin head on this sarcomere, they're both, if both sarcomeres contract, are going to be at that Z disc. Therefore, the I band will be very, very small. Okay? Okay. Again, maybe challenging to conceptualize on your first time through. I want to encourage you to pause, come back to this, and draw out what it actually looks like, okay? Here is some point-by-point descriptions of what shortens in the sarcomere, as I just described it. And then the opposite, although I didn't go through that process, will happen as the sarcomere returns to its original length and looks something like that, okay? So again, draw out sarcomere at relaxation, partial shortened, completely shortened, and maybe even slightly overstretched or relaxed. Okay. So that covers the main contractile fibers, is what we call them. So the fibers that are involved in contracting. So this myosin thick filament, this actin thin filament. But we notice that there's other proteins kind of woven around, like this yellow one, some of the purple around these other filaments. And let's explore what those are, because they're really important to the structure and contraction of muscle. Okay? What we call these are non-contractile proteins. So these proteins don't directly or aren't directly involved in the contracting muscle. Okay, but what do they do is provide structure and strength to the sarcomere. Okay. And some elastic components as well to the muscle fiber, as we'll see. So again, let's take a little look here at the structure of the non-contractile proteins. Where do we find them? And I feel this helps us understand a little bit their purpose. So again, we find titin here, and what its main goal is to add some structure and stability by anchoring myosin, okay? So it connects the Z line here, or Z disc, to the M line, essentially, by reaching out to myosin along the longitudinal axis of the sarcomere. Okay. It adds some passive stiffness and limits range of motion to a certain degree. Now the other part that we want to check out here is nebulin. So it's the purple line along the sarcomere here. Okay, and it's primarily localized to the I band, right, to the section here, but as you notice, it stabilizes actin. That's its primary role. Okay. Okay. So if we want to explore how contraction works a little bit, we need to dive into this component of our actin filaments, because they're active, let's say. All right. We need to speak about actin because it is one of the major components as far as allowing cross-bridge or attachment sites to occur, to allow myosin, that thick filament, to connect and paddle along or pull along the thin filaments. Okay, and that's largely done by these regulatory protein molecules called tropomyosin and troponin. We'll explain how they work exactly in the next section. But what we'll find is that there's these three subunits that hold on to the actin, or the thin filament. They hold on to tropomyosin, which is this other band we'll see in an image in a moment. And then troponin C binds calcium, which has to do with the activity of creating force. Okay. So again, if we look here at actin, thin filament, we have the red, which is our actin. So they're just these this helixy wound protein. Okay. But then around it, we have this called tropomyosin. Okay. This is like another strap or a band that goes around tropomyosin. And what will happen is we'll see this covered site of these regulatory proteins in one position, but as we're ready to undergo a contraction, muscle contraction, what will happen is our troponin active site will be exposed and allow calcium to bind here, and that's a big thing. So here it can't bind, it's not exposed. Here it is. We can see how our one troponin subunit of this regulatory protein, so the purple here, holds on to the actin. This component up here holds on to troponin, so it's kind of anchored on the two. And then we have our active site where calcium can bind. Okay. Yes, that would be troponin. And then to look at myosin, again, these are our contractile proteins. Contractile, our non-contractile is nebulin and titin. Okay. Just to try to categorize those in your mind. So our myosin filament has those globular heads I mentioned, like the paddles, to help row along. The heads are made up of an enzyme, myosin ATPase. We're going to explain that role next up, but it's, as you can imagine, has to do with ATP. And the tails intertwine essentially to form the myosin filament. Cross bridges are important when we talk about contraction and generating force. You'll see that the globular heads interact with actin, as I've been describing, and pulls them along to create contraction between the thick and thin filaments. Okay, so again, just looking at this thick filament, you can see there's lots of globular heads on the structure, and all the tails are wound together to make the cable part of myosin. Okay. So yeah, I already gave a little review, but here we are again. So we got actin, thin filaments, and myosin, thick filaments here that interact together to cause movements. We have titin and nebulin, which provide some structure to the cell. And not pictured here would be tropomyosin and the active site that winds around the actin filaments. Okay. Next up is the sliding filament theory, and that will help explain how our muscles actually contract.